Hello, knowledge seekers. Welcome to another session of First a Scientist, where we will be discussing all the same topic, metabolism of carbohydrates. Till yesterday, we have discussed topics covering like, basics of what are carbohydrates, what are the rich source of carbohydrates, why do we need carbohydrates. So, in the metabolism of carbohydrates, what are the processes involved, of which glycolysis, very first step, which is a very much important process in metabolism of carbohydrates. So we've studied various reactions involved in glycolysis and various and what are the how the energy is liberated during this glycolysis. What is uh, lactic acidosis and its significance. We have studied various topics. Yesterday we have also learned about overall ATP overall number of ATP synthesized uh, because of the reactions or because of the products that are produced from glycolysis. Today we'll be looking at two more topics. So one is glycolysis and shuttle pathways, and the other topic we'll be discussing is cancer and glycolysis. So let us dive into our first topic today, which is glycolysis and shuttle pathways. So so we know that shuttle pathways, we majorly study this topic in biological oxidation process, biological oxidation. So what does this shuttle pathways basically mean? So you know, right, shuttle, um, to and fro motion between a to and fro motion between two fixed points can be referred to as a shuttle, to and from motion. That's what basically shuttle refers to. So let us look what is the significance of glycolysis and shuttle pathways. So the reduction equivalents, which are nothing but NADH produced in the glycolysis, when get involved in the shuttle pathways, we see an increase in the we see an increase in the production of ATP by a margin of 3 or 2. So now let us understand more deeper. So what are the various shuttle pathways where glycolysis has its role? So so there are basically two pathways where glycolysis role is directly involved. So one is aspartate and malate shuttle pathway, and the other is glycerol phosphate pathway. So where do these occur basically? So in these two occur under aerobic conditions only. The As I'm quoting the word aerobic condition, that means there is the involvement of mitochondria for these pathways. So now let us understand more deeper about these pathways. So basically, the mitochondria is a double-layered, double-membraned organelle present in the cell. So double membrane organelle in the sense it has two membranes. One is the outer membrane, and the other is inner membrane. So the outer is called outer mitochondrial membrane (OMM), and the inner mitochondrial membrane is abbreviated as IMM. So basically, these reduction equivalents, which are nothing but NADH or FADH, so they are impermeable, highly impermeable to the uh, inner mitochondrial membrane. So the outer mitochondrial membrane is relatively permeable to these heavy molecular weight compounds, whereas the inner mitochondrial membrane is almost zero permeable to polarized ions. So that's why it's very hard for these uh, mm, reduction equivalents to pass through the barrier, that is the inner mitochondrial membrane. So what happens here is this. mm so now so these reduction equivalents are transported into the matrix or into the inner mitochondria, into the intermembranous space of mitochondria by two different pathways, which are nothing but which are actually called as shuttle pathways. So the pathways through which these reduction equivalents get transferred or get transferred into either mitochondrial matrix or intermembranous space are called shuttle pathways. So what are the two various shuttle pathways involved? As we discussed, one is glycerol phosphate and the other is uh, aspartate malate pathway. So in this glycerol phosphate pathway, what happens? Let us look at the steps involved in and the constituents involved in this glycerol phosphate. So basically, what happens is, so the NADH produced in the reaction, in the sixth reaction, which is nothing but oxidation reaction, where G3P is oxidized to 1,3-bisphosphoglycerate by production of NADH2. So this NADH2 enters into this shuttle pathway, which is glycerol phosphate pathway. So what happens is, in this glycerol phosphate pathway, NADH undergoes oxidation under the action of catalytic action of enzyme glycerol phosphate dehydrogenase. That too precisely cytosolic glycerol phosphate dehydrogenase enzyme. Why am I referring it to cytosolic enzyme? Because it is present in the cytosol. So in cytosol, this NADH2 undergoes oxidation. That means, to put in another way, it undergoes dehydrogenation. That means it loses a hydrogen molecule um, under the catalytic action of enzyme cytosolic glycerol phosphate dehydrogenase. So to which substrate does this NADH loses its hydrogen molecule? So glycerol phosphate is glycerol phosphate, DHAP, dihydroxyacetone phosphate. It is present in cytosol. This takes up that hydrogen from NADH and undergoes a reduction to form glycerol 3 phosphate. So DHAP is impermeable to the membrane of mitochondria. So this DHAP is now reduced to glycerol 3 phosphate, which is more permeable to mitochondrial membrane. So this glycerol 3 phosphate enters into mitochondria, enters into the mitochondria. So in the matrix, so this G3P, which is not G3P, which is glycerol 3 for glycerol 3 phosphate, undergoes an oxidation reaction. Undergoes an oxidation reaction. If you remember, so there DHAP had undergone a reduction reaction. NADH has has lost its hydrogen molecule to DHAP to convert this DHAP to glycerol 3 phosphate. So this glycerol 3 phosphate in mitochondrial matrix undergoes oxidation reaction, losing its hydrogen to reduce FAD to FADH2, and this FADH2 produced in this reaction enters into electron transport chain and results in production of two ATP molecules and results in production of two ATP molecules. Remember this very clearly. So and in the same way, after losing a hydrogen molecule, the glycerol phosphate is again converted back to DHAP under the same catalytic action, activity of enzyme mitochondrial glycerol phosphate dehydrogenase. It's very simple. See, as it is present in the mitochondrial matrix, it is called as mitochondrial glycerol phosphate dehydrogenase enzyme. So this DHAP, which is eventually produced, is uh, again sent back to cytosol, and the shuttle pathway is again reinitiated. So this is how one shuttle pathway completes and another shuttle pathway continues. This reaction continuously happens. So from the shuttle pathway of glycerol phosphate, we get two ATP molecules produced. So this is one pathway. Now let us look into the other pathway of in shuttle pathways, which is aspartate malate pathway. So in this aspartate malate pathway, see, understand this very clearly. What happens is, see, in cytosol we have this a 4-carbon compound called oxaloacetate. So this oxaloacetate is highly impermeable for the internal membrane of mitochondria. That means it cannot pass through the internal membrane of mitochondria. So what happens is, so remember very clearly, this particular pathway occurs always in the intermembranal space. So what happens is, this oxaloacetate undergoes so this oxaloacetate undergoes a reduction reaction in the presence of NADH to form malate, so which is a permeable compound. So this it can just cross the outer mitochondrial membrane and enter into the intermitochondrial space, intermembranous space of mitochondria. So this malleate enters into the intermembranous space, where due to the activity of enzyme present over the outer surface of inner mitochondrial membrane. See, remember this very clearly, understand this very clearly. So an enzyme is present over the surface of inner mitochondrial membrane. That means it is facing towards cytosol. So as the malleate enters into the intermembranal space, so this enzyme gets activated and it uh, oxidizes malleate to oxalate, so resulting in the reduction of NADH, sorry, NAD to NADH2. So this is what happens. So the reduced NADH2 is transported to electron transport chain, where it results in production of 3 ATP. So you remember, you understand this, right? So from aspartate malate pathway, we get 3 ATP synthesized as the reduction equivalence enters into the electron transport chain. So that is how it is. So the oxalate formed here now undergoes a transamination reaction in the presence of enzyme aminotransferase. So along with glutamate. So this oxalate and glutamate together undergoes a transamination reaction under the presence of aminotransferase enzyme and results in the formation of aspartate and alpha ketoglutarate. So aspartate and alpha ketoglutarate, they both are expelled into the cytosolic membrane, where they both again undergo transamination reaction, eventually resulting in the formation of oxalate and glutamate back to normal compounds, so back to the original forms, glutamate, under the catalytic activity of aminotransferase present in the cytosol. So we now have oxalate, with which the shuttle pathway is actually initiated, and the glutamate enters back into the mitochondrial intermembranous space of mitochondria. So this is how this shuttle pathway repeats. So we have to understand, to quote the catalytic so the catalysis of um, oxalate to malleate is driven by the enzyme, is driven by the enzyme malleate dehydrogenase. So malleate dehydrogenase is the enzyme which catalyzes the reaction of conversion of oxalate to malleate by the oxidation of NADH. So there, from NADH, the reduction equivalents are taken up by the oxalate to form malleate. So this malleate enters into the intermembranous space, where we have malleate dehydrogenase stick to the outer surface of inner mitochondrial membrane. So this entire process occurs from cytosol to intermembranous space. So this is what aspartate malleate pathway. So this is aspartate malleate shuttle pathway, and this results in the production of 3 ATP molecules when linked to electron transport chain. So these are the two pathways which have the involvement of glycolysis. So now let us now logically answer some questions. Um, and that is, see, it's very simple. So we've understood what is aspartate malate shuttle pathway, and we have understood so glycerol phosphate shuttle pathway. So what is the basic difference between this, so the glycerol 3 phosphate that we have studied in glycerol phosphate pathway, and the glyceraldehyde 3 phosphate, also which we have studied in glycolysis reactions. So it's very simple. See, so glycerol 3 phosphate is actually formed from the glycerol under the action. So it's very different. So glycerol 3 phosphate is formed from the, so glycerol by under the catalytic activity of glycerol kinase can be formed from glycerol kinase. That means that is a general reaction of glycerol 3 phosphate. So whereas glycerol 3 glyceraldehyde 3 phosphate is formed from DHAP through an rearrangement reaction. Here, so from DHAP by a reduction process. I mean, DHAP here is reduced to give rise to glycerol 3 phosphate. So there is a different reaction. So DHAP, when undergoes a rearrangement reaction, results in the formation of glyceraldehyde 3 phosphate, whereas DHAP, when undergone reduction reaction, it results in the formation of glycerol 3 phosphate. So this is the major difference. You have to understand it very clearly. So in the naming itself is very clear. So glycerol contains hydroxyl group, whereas glyceraldehyde contains an aldehyde aldehyde group in that. So now let us look at now understand the let us try to understand glycerol phosphate pathway with uh, with with a deeper question. So which is the reducing equivalent of NADH in glycerol phosphate shuttle pathway? So you understand it. See, basically NADH are the reduction in equivalents involved here, as they directly cannot enter into mitochondria to and so that they can enter into the electron transport chain. So they are they are generating here the reduction equivalents to carry forward these hydrogen molecules so that they can enter into the electron transport chain. So what is the actual reduction equivalent for NADH in glycerol phosphate pathway? So obviously the answer would be um, so which is just take a step back and understand. So in this in the first reaction of glycerol phosphate shuttle pathway, which molecule is reduced? So DHAP is reduced to form G glycerol 3 phosphate. So that means glycerol 3 phosphate is the reduction equivalent of NADH in glycerol phosphate shuttle pathway. So where does glycerol phosphate shuttle pathway occur? So where does this occur? Understand this bit. As I told you, so mention so glycerol phosphate shuttle pathway also occurs in, so it occurs in intermembranous space, but not in the matrix of in the mitochondrial matrix. So uh, if I remember, I was telling you that this reaction occurs in the matrix, but a correction that this doesn't occur there. It actually occurs in the intermembranous space of mitochondria. That means between the two membranes, which are outer membrane and the inner membrane. So I'll be sharing so in the description of the video there's a link where you can actually open and understand. So where does this glucose phosphate pathway actually occurs? So this is very important to understand, as the enzyme slow glycerol 3 phosphate glycerol 3 phosphate dehydrogenase is present, or it is stick to the outer surface of inner mitochondrial membrane. It occurs, this entire shuttle pathway occurs in the intermembranous space. So we have to be clear on that. So there is a document which you can access to look at it. So in many literature books, so though this is clearly mentioned, the illustrations or the diagrams are a bit confused, I mean create a bit confusion. So this article would be very clear for you to understand that. So this occurs in these shuttle pathways occurs in the intermembranous space only. So now the another question is like, which is the reducing equivalent of NADH in aspartate malate shuttle pathway? Is it oxalate or malate? So remember correct. Oxalate undergoes reduction and results in the formation of malate. Thus malate is the reduction equivalent of NADH in aspartate malate shuttle pathway. Okay. So you're clear. So that is also very clear. Now understand like which of the, let me give you another question like which of the following is correct? Like so malleate, the inward movement of malleate occurs only if the outward movement of aspartate is present. So that is, so I'll be giving you two statements. Okay, so the inward movement of malleate occurs only if there is an outward movement of aspartate. That is statement one, and the other is next is statement two, which reads like this. So aspartate outward movement occurs in complementary with the outward movement of alpha ketoglutarate. Out of these two statements, which is right? So if you understand the reaction clearly, so oxalate undergoes a reduction and and turns into malate, and malate is taken into the malate enters into the intermembrane space. So after their malate undergoes oxidation reaction, which results in the formation of oxaloacetate again. So this oxaloacetate, in the presence of glutamate, undergoes a transamination reaction, so which results in the formation of aspartate and alpha ketoglutarate, which are permeable to, or which are eligible for the outward movement from the mitochondrial membrane, and hence they go out. So here, so if you understand clearly, so the movement of oxaloacetate, so oxaloacetate cannot enter into the cannot cross the mitochondrial membrane, but the oxaloacetate present in, that means it is same. So once the that means the oxaloacetate present in the intermembranous space also cannot cross the cannot go out. That means its movement is unidirectional. That too, oxaloacetate only after getting reduced to malate can enter here. So again, you don't observe malate going out. You observe a different molecule, malate turning back to oxaloacetate, and this oxaloacetate getting undergoing a transformation reaction and forming aspartate, and this aspartate is going. So in the entire this process, you don't observe any complementary between the inward movement of malate and the outward movement of aspartate. But so glutamate and oxaloacetate undergoes a transamination reaction, transamination undergoes a transamination reaction, resulting in the formation of aspartate and alpha ketoglutarate, and these both are taken out of the mitochondrial membrane. That means the outward movement of aspartate is in complementary to the outward movement of alpha ketoglutarate. Thus, second statement 2 is correct here. So this is basically the significance of glycolysis and its involvement in shuttle pathways. So it's very simple. So glucose phosphate shuttle pathway. So glucose phosphate shuttle pathway so results in mm, production of 3 ATP, whereas sorry glucose phosphate shuttle pathway results in production of 2 ATP, whereas uh, aspartate malate pathway results in production of 3 ATP. That's very simple. Now let us look at cancer and its glycolysis and its relevance in cancer cells. So what happens is basically, so cancer cells naturally consumes uh, more more glucose, and that means there is an increase in uptake of glucose in cancer cells, and thus glycolysis also occurs at a higher rate in cancer cells. So also the one thing is like cancer cells are devoid of and this results in the less supply of oxygen by the blood vessels to the cancer cells. As cancer cells results in formation of tumor, it becomes hard for the blood vessels to supply oxygen to the cells in that tumor. So this results in a formation of a hypoxic state in the cancer cells. That means oxygen levels are reduced. So cancer cells adapt to a condition where glycolysis occurs in a hypoxic condition, in a hypoxia, in the state of hypoxia, or in a hypoxic condition only, due to the hypoxia induced transcription factor, hypoxia induced transcriptional factor. So and the glycolysis occurs continuously under reduced oxygen state only. But the thing is, the cancer cells cannot survive longer periods so without oxygen. So that this in the treatment of cancer cells also, in the treatment of cancers also, so one strong a way which is still followed in clinical practice is that so inducing a kind of uh, giving drugs or taking the patient under medication where mm, these drugs actually induce a induce a state of less vascularization for the tumor, so that with more hypoxic condition the cancer cell eventually dies. So these are the topics for today. So we have seen various shuttle pathways and glycolysis, and also we have seen the glycolysis and cancer cells. So now let us look at the other topics tomorrow in the video. So don't forget to subscribe to my channel. Please do like and share your comments, or if you have any doubts, I'll be very happy to answer all of them. So thank you for watching my video. Till then, see you.